So this talk is going to be a largely historical talk, ending over 24 years ago, but we'll get going. So let's see. I'm trying to move. Okay. So the problem of protein and DNA sequence comparison, I tend to think as having sort of three main components. The first is defining exactly what it is that you're looking for, and this varies, of course, from one application to another. Once you've settled on a definition, you then need to figure out how do you find it, which is essentially an algorithmic or computer science problem. And then one's usually faced with the question, well, now I found it, does it mean anything? And certainly statistics has a lot to say to that. Now, in the early days of sequence comparison, there were no DNA sequences. They only started coming in the 1970s. One was dealing with proteins. And the earliest protein sequences one had that were related globally. I don't know that this particular example that these proteins were sequenced early on, but this is an example of two related sequences that were globally related. So the definition, original definition, was for global alignment with the idea that one could insert gaps or null characters into either sequence to try to line them up as well as possible. That all of the letters would participate. There wouldn't be any crossing over. You wouldn't align letters in the same order that they occurred in the two sequences. And there would be some kind of score that you would define that you would try to optimize. And the early scores were based basically on matches, mismatches, and gaps, but got elaborated into more fancy scoring systems after that. For the global alignment problem, the first really published algorithm in this field was that of Needleman-Wunsch in 1970. And they observed that if you had a structure like this, which is called a path graph, you wrote one sequence down the side, one across the bottom, then a path starting at the upper left and ending at the lower right would correspond to a global alignment. And you could change the problem into what is the best scoring path through this path graph. Now, as people started sequencing more proteins, they discovered some that actually seemed to be related over parts, but were not globally related. This was not obvious that this was a property of proteins early on, but we came to understand that proteins have what are called domains. And so in this sort of graph, each of these colored regions corresponds to a domain that can be shared with different proteins. And you could have a domain at the beginning of one protein that occurs at the end of another in more than one copy. So in the path graph terminology, that this region might be related to these two regions, but nothing else would be related. And what one was actually looking for were these sort of little diagonals within the path graph. This led to a redefinition of the problem, looking for local alignments. And the first real, the definition of the problem that eventually got accepted was that published in '81 by Smith and Waterman, and the algorithm needed to change as well, but it still operated in order mn time, basically the area of the path graph. Now, as sequences, protein sequences, and then later DNA sequences were being found, they were built into databases, and so one had the problem of having one sequence that you weren't comparing to one sequence, but to a large database of sequences, which you could think of as just one very long sequence where I've strung them together, separated by breaks. The time that it took to search these databases using this Smith-Waterman algorithm was a long period of time. It would take about an hour to do, and people wanted to do this a lot faster. And so a number of fast heuristic algorithms were developed during the 1980s to do these database searches, heuristic in that they were not guaranteed to find you the best solution, but had a very good chance of doing so. You know, in a database search, one might be looking for just this one similarity, this match to one sequence in the database, and all the time that you're spending searching all the rest of the database is sort of wasted, and so one wanted to speed things up. You'll notice a common feature, local alignment of these three citations is Lippman DJ, that's David Lippman, who was a prime mover in this field, and I went to work with him as a postdoc in 1987 at the National Institutes of Health. Now, in the mid-80s, the idea of sequencing the human genome was first proposed. This is perhaps the first kind of public proposal for this in 1986. This was debated by various panels for several years, but and the project was officially launched in 1990, although it was definitely in the works before then. Congress, US Congress noticed this and felt that there was going to be a need for government center focused on basically what we now call bioinformatics. And in the November of '88 passed a public law, was signed into law creating the National Center for Biotechnology Information. In the early part of 1989, the leadership of NCBI was appointed. This included David Lippman was the director, Jim Ostell, who was in charge of software development, and Dennis Benson, who made sure that everything ran properly. So I, being a postdoc with David Lippman, moved over there with him in early '89. And in the spring of '89, we started getting visits to this new center from a number of friends. These included Sam Carlin, a mathematician at Stanford, and I'll have more to say about him. Webb Miller, a computer scientist at Penn State, and Gene Myers, computer science at University of Arizona. These two had collaborated on quite a number of papers in bioinformatics before this, and were good friends. When while Gene was visiting, or just perhaps the week before, a news article appeared in Science describing a new computer chip and its application to finding patterns in biological sequences. And Gene and David Lippman, being much more software than hardware people, felt that this article included a bit of hype, and they were thinking we can certainly do as well as this chip in software. So one evening, while Gene was having dinner at David's house, they had the spark for BLAST. And the basic idea is that computer science provides very, very fast, rigorous algorithms for finding perfect matches, basically by indexing things and looking them up. So why you can search the whole web on Google in a fraction of a second. The problem with, certainly with protein sequences, is that frequently significant similarities between two proteins may not share more than two identical letters in a row. But the idea was that maybe anything that was strong enough to actually be a biological in significance would contain short words that could be matched and could be found quickly and would be a hook to find those alignments. And so this is the BLAST search algorithm in sort of one slide, at least that applied to proteins. What one was looking for was something like this, which we called a high-scoring segment pair. The original BLAST program did not allow any gaps. It was simply looking for ungapped local alignments. And you'll notice that this high-scoring segment pair, which maybe is statistically significant in a database search, doesn't have any more than 2 letters in a row identical. But the idea is that using a standard scoring system such as Blossom 62, it might contain word pairs, which we would call hits, that would allow you to find this. And so the idea is that you would take the query sequence, you would have some threshold score, and for each word, I'm just illustrating this word, but for each word you would determine every possible hit to this that had a score above a threshold, here T13, you do that for each word, and then you'd scan the database, and whenever you found a hit, you would look in both directions and see whether it was part of an alignment that had a high enough score to be interesting. Here, you can imagine there being a hit inside of each of these diagonals. The problem is that if you extend this as far as possible in both directions, most of these hits are completely random. You're going to be wasting a lot of time. So they had the idea that we'll search until the score drops by some fixed amount, amount X, which we'll define. And if the best score drops more than X beyond what we found before, we give up. So this way, we don't spend a lot of time searching for insignificant hits. Here, we might get a hit with a high score that was statistically significant, and here another one. This is of course a heuristic in that you may give up after you've extended a certain amount, and if you had extended farther, you might pick up another match here that would actually increase the score. So this is a heuristic algorithm. Now, Gene Myers was more interested in extending this idea in a way to make this a rigorous approach, whereas David Lippman was happy to have this as a very fast heuristic method. And so they kind of diverged, and David wrote in Webb Miller to write the original BLAST code. As this was being, and here is basically a slide showing that there is, of course, this trade-off in speed. If this is a T13, this is the amount of time it takes to run in seconds. As you increase the threshold, you had fewer words that you had to look up. And as you increase the threshold more, the program would run more quickly. But of course, this also led to a trade-off in sensitivity. For a particular threshold, this shows the probability of missing or log probability of missing an alignment depending on the score. The higher the score, the less likely you are to miss it, and the lower the score, the more likely you are to miss it. A couple of collaborators were then added to the project. I came in really to work mainly on the statistics involved, basically, first of all, the statistics of the parameter settings, and also the statistics of the database search results, which I'll have a little bit more to say about. Warren Gish introduced a faster way of searching for these matches and rewrote the code so that the first production code was code that Warren had written. Just to say something about my own contribution here, back in 1985, it was observed by Smith, Waterman, and Burks that for the sorts of local similarities people were looking at, when you're comparing random sequences, they tended to grow as the log of the product of the lengths of the sequences compared. The first analytic result that was analogous to this was an asymptotic formula for the longest run of matches with exactly k mismatches. Now, this is very interesting from a mathematical standpoint but that kind of scoring system was not at all what people wanted to use. They weren't looking for longest run with exactly a certain number of mismatches. They wanted to use scores that better captured the relationship among the various amino acids. I had a conjecture about this, which I brought to Sam Carlin when he actually, it was before he visited. I brought it to him in the previous fall, but when he came to visit, he had worked through this conjecture, and this led to the publication of several papers here. This is the first description of analytic statistics for alignments using general scoring systems, but also without gaps. This is exactly the kind of alignment that BLAST was going to be producing, so that from the very beginning, we could put numbers on whether the alignments it found were statistically significant. Just to say a couple things more about this paper, it really had two main elements. The first one was a formula for how many alignments with a certain score you expected to find. So the number that you expected to find purely by chance was a decreasing function of the score, and all of the parameters here had analytic formulas. We also showed that the optimal scores to use depended on if you were looking for alignments with certain background probabilities of various pairs of sequences being matched, that these were the optimal scores to use. So, for example, these are all sort of true optimal alignments. Well, I don't know that these are optimal, but these are alignments representing true relationships between human beta globin and globins from different species. You'll see, of course, that the frequencies of matching letters and of various mismatch pairs of letters is quite different as one moves from one to another, and therefore these scoring systems that are tailored to best find these vary as the sequences get more and more distant. We had been calling this program BLAST for a while because it rhymed with the fast from FASTA, and it sounded like it was a lot faster, which it was. But I woke up one morning and realized that BLAST was actually an acronym for Basic Local Alignment Search Tool. So the word now had a meaning. In the fall of '89, we submitted it to Science. It was rejected by Science. We made maybe a few minor changes, submitted it to the Journal of Molecular Biology, which agreed to publish it in 1990. And I guess this came out in the fall of 1990. Now, BLAST was originally not really meant to compete with FASTA. The idea was it was simply going to try to find things that were obviously related very, very quickly. But it worked so well that people started using it in lieu of FASTA. And so to compare it to the program, it had a major disadvantage to FASTA in that it only produced alignments without gaps. This was in 1990. It was not until 96 and 97 that versions of the program that allowed gaps were introduced. The major advantages it had is that it was a good deal faster and it had available statistics. And the other really key advantage it had was that it was developed at NCBI and embedded in a larger system called Entree. So to take you back to the mid-'80s, by then DNA sequences had begun to appear. There was a database of DNA sequences called GenBank. And here is GenBank in 1984. Here is GenBank in 1985. Here is GenBank in 1986-87. As you can tell, this idea does not scale. And I think this may be the last paper version of GenBank that was published. In the early 1990s, NCBI, which took over the management of GenBank not long after it was established, released the first version of GenBank on CD, but it included with this a whole new set of programs that link the DNA and protein sequences. If you had a DNA sequence and there was a coding region, you could go to the protein sequence and link both of these to the Medline abstracts. It compared these to one another using BLAST in this case, and other people at NCBI had developed methods for comparing abstracts to say, show me a paper that is like this. Before this, you would get a search, you'd go down to the stacks, pull out the paper, find the paper, maybe pull out the sequence, run it through some program to give you the protein sequence if it was a DNA sequence and so forth. But this was all now automatic. And this expanded as more data became available in 1994. Structural similarities were added. By 1996, we began to have genomes of simple organisms. And so the fact that BLAST was actually integrated into this much larger system, I think, has to be one of the great reasons for its success. If it had simply been an academic program that ran faster and was not integrated into this larger system, it would not have had the same impact. And furthermore, Medline was a subscription service in 1996. I'll get to that in a moment. Here, by the way, are the people at NCBI who had developed this whole system. This is on a little picnic. Here's David Lippman. And in 1997, for the first time, Medline became completely free and open to the public. Here we have Don Lindbergh, the director of the NLM. You have the director of the NIH, the director of the NCBI meeting, announcing this with Al Gore and making this all available to the public. So that is the same year, by the way, 97, we released a version of BLAST that did allow gaps and was faster as well. And that I will not talk about here. So that is a brief history of the development of BLAST, and I'll end it there.
Thank you so much, Stephen. This was really interesting. I suppose my first question is really going to be, about the timelines here. So it took 6 years to introduce gaps into BLAST. I'm wondering, was it really just because this was a difficult feature to implement from the sort of technical point of view, or was it more sort of like a decision led by what the users actually needed and what the users wanted?
I think that it did actually take us a while to figure out how to do this in a rapid way. The basic, and we did need to develop another number of ideas on how to do this. The paper describing this in 97, you'll see that there are actually quite a number of ideas that had to be introduced. In the meantime, we had actually gotten around not having gaps by working out the statistics of multiple ungapped alignments, so that a gapped alignment, you can just show, you can break it into the ungapped parts, and if each of those is large enough, you could report it that way. So that was one of the early changes in BLAST. You'd run this and you might get an alignment, and you'd get actually 3 or 4 hits that might clearly be part of a larger alignment, which we could come up with a statistical assessment of finding multiple hits to the same sequence. So that was one element of pushing it. We had some ideas of how to introduce gaps, but I guess it just took us a while to get around to implementing it.
I see. Yeah, that makes a lot of sense. And my other question is really just about kind of biological significance of mutations, because BLAST can deal with sequences which are mutated, which are diverged, if you like. But it's going to look at the the sequence at the DNA level. What about differences that actually are coming? What about mutations that do change the DNA, but actually do not change the protein? They create a different triplet that would actually encode the same amino acid. So was that taken into an account as well at some stage?
Well, first of all, when you have a DNA sequence that codes for a protein, you can either be comparing, if you're trying to compare it to other sequences, you can compare it to other DNA sequences, or you can translate the sequences to protein and compare the protein sequences. So for instance, if you had a mutation that was a silent mutation, once you translated it, it wouldn't show up there at all. There's sort of a rule of thumb that people have used that says if it translates if it encodes protein, it's always better to compare it at the protein level. That's actually not quite true, but it's a sort of a subtle question. I did publish not a very widely known paper on this. It's States et al. That I recommend to people to understand the idea. People were saying you're throwing away information when you translate it to it seems odd that if you translate it to protein and you throw away information by doing that you actually get a more sensitive search. What people are really talking about are naive comparisons of DNA where you're not recognizing the genetic code. And the genetic code is a key thing of how to score things the best way. So in general, it is true. If it codes for protein, you don't want to be comparing DNA. You want to be translating it and comparing the protein, though that's a sort of a subtle question of the details of why that is.
I see. My other question is really just about, not so much about the past, but about the present and actually the future, because sequence alignment has taken us incredibly far. So, just recently, DeepMind has broken huge news. They were actually able to achieve incredible accuracy of predictions of protein shape using approaches. Obviously, they were not mathematical or statistical approaches. These were more sort of machine learning or slightly more black box sort of approaches that were able to achieve that. But did you see this coming at any stage, Steve, or was it was it really something that came out of the blue?
Was certainly the AI applications have not been something that I had paid that much attention to, although I've heard about it more through the popular press, and this, of course, has been extremely exciting developments. I think that people can do, of course, a lot more. You can find out a lot more than you can find with BLAST. One's using a very limited amount of information of simply this linear information. It doesn't have anything to do with interactions and so forth. And so people, of course, in developing SciBLAST, we're using multiple alignments, but once you add in information from structures and so forth, you can try to capture, there's all this information that about the biology that's implicitly there that BLAST doesn't have access to. But most of these other programs sort of use BLAST as a foundation to build other things on top of. And so that's one way that it remains relevant.
Okay, Stephen, this is a great perspective, and thank you so much for teaching us about the history of BLAST. it's something that I listened to with really great interest. Thank you so much for taking the time and coming to the conference.
Thank you.